Good evening. Welcome to March's Evening at Whitney lecture. Glad that you're all here. Before I introduce tonight's speaker, I would like to encourage you to come back in a month. In April, we'll have another free lecture, and it will be about the biology of aging from faculty coming from Gainesville at UF. Okay, I guess one thing I forgot to say is introducing myself. I'm Elaine Seaver, a professor of biology based out here at Whitney Lab, and it's my real pleasure to introduce tonight's speaker, Dr. Paula Murphy, who is a professor of zoology, University of Dublin, Trinity College of Dublin, and she actually just made the trip a couple of days ago from Dublin. We don't typically have, as part of this series, international speakers, but we're very fortunate to have Dr. Murphy. I'm really excited to be able to introduce her because she works in the field of embryogenesis, which is my chosen field of study, a field where we're trying to understand how cells become different from each other during embryogenesis and how all the organ systems form properly during the process of development. Okay, so let's me stop talking. Dr. Murphy is originally from Ireland. She did her training is actually quite international. She started off at the University of Dublin, where she is now, as an undergraduate, but then went to the University of Edinburgh in Scotland for her PhD, and then did a few postdoc sequentially, first in Rome, then Paris, and then I guess had a research position in Oslo, and then eventually joined the faculty back in Dublin. So she has quite the international experience. And I'm not going to go on any longer. I'm very much looking forward to hearing her tell us about development in invertebrate embryos. Dr. Murphy.
Good evening, everyone. Yeah, the mic's on, I can hear it. And thanks very much, Elaine, for that lovely introduction. And it's such a great pleasure to be here and have a little break from the cold, dark winter in Dublin and be here at the Whitney Laboratory, which is a fantastic location. And I'm so looking forward to interacting with everyone here more tomorrow as well. So as Elaine said, I'm at Trinity College in Dublin, and we have a lovely old historic campus right in the centre of the city. And if you haven't already been there, I'd urge you to visit because it's a relatively calm oasis in the middle of a very busy city. As Elaine said, I'm also a developmental biologist and interested in how, focusing more on vertebrate animals, we use the chick and the mouse as our model systems in the lab and looking at how vertebrate animals form. And I'm going to give you a very broad introduction now to some of the questions that developmental biologists like to research. And these questions really stem from one phenomenal fact, and that is that a single cell, a fertilized egg cell, can give rise to a complex multicellular organism. So even an organism as complex as this one, or you or I, started out as a single cell. And that single cell, and this is a beautiful photomicrograph just at the point of fertilization. So those two objects you can see in the center are the pronucleus of the egg and the sperm as they're about to fuse to generate the new genetic identity of the individual. And that single cell has to give rise to all of the billions of cells in our adult bodies or in the adult form of other vertebrate animals. And all the hundreds of different cell types. So the muscle, the blood, the neurons in the brain, the bone, all very specialized cell types that have to derive from this single cell. So that's one of the key questions we ask as developmental biologists. How do cells differentiate from that one cell to form all these different cell types with their very specific characteristics? And another big question arises from the fact that these different cell types as they emerge are very tightly regulated in temporal and spatial, in the time and space in which they form, to form a structure like a hand, with, you know, the bone and the muscle and the blood and the neurons in there, but arranged in a special spatial arrangement. And so the other big question that we're interested in is, how is the body plan organized? How are these different cell types arranged to form structures and organs? And of course, we study what's happening in the very early embryo as all this is being set up. So we're focusing on how all of this organization is set up in the early embryo. So, as you've just heard, this single cell has to give rise to all these different cell types, the muscle cells that will cooperate together to form muscle tissue, the neurons that will form interlinked connections for communication, and connective tissue. So these cells in connective tissue, they construct a very elaborate extracellular structures to make tissues like bone and cartilage and tendon. And these different cell types emerge over time in the embryo. They're differentiated over time. And then these different cell types and the tissues that they form have to be organized spatially to form structures and spatial organization of the emerging cells and tissues. And the images you see here are from our lab, and these are images of a very tiny mouse embryo limb as it's forming. You see the scale bar here, this is really tiny, but even at this stage, we can stain for the appearance of different tissue types. So blue here stains cartilage, red stains bone, fluorescent red stains muscle, and fluorescent green tendon. So even this tiny little emerging limb, the cells are being organized spatially to form a mouse front leg. And those spatial arrangements are species specific and arranged differently in different species. So as this image taken from a review here very nicely shows the equivalent structures across the forelimbs of different vertebrate animals. The blue is the humerus, the green is the radius and ulna, the yellow, the carpals and metacarpals, and the brown phalanges. So different spatial arrangements of these corresponding parts of the skeleton make the difference between a human arm or a bat wing or a dolphin flipper. So this spatial arrangement is very important. Now here's a quote from a wonderful Czech poet and immunologist, Miroslav Holub. And in 1990, I was writing my PhD thesis, and I happened to be listening to BBC Radio 4, and there was a panel discussion. And together with Miroslav Holub, there was a physicist and a computer scientist. And the gist of the conversation was about the future of space travel and virtual reality. And it was sort of saying how great us humans are and what incredible things we can do. And Miroslav Holub wasn't saying very much, and the chair asked him to comment. And he spoke very slowly and in a heavy Czech accent, said that mouse embryonic development is like turning a lump of iron ore into a space shuttle overnight. And I got from that the inspiration to continue writing my PhD thesis. And I've never forgotten it. So getting back to this single cell then, that has to form that whole complex organism, depending on the genome inside that cell can give rise to a human or a mouse or a frog or a fish. So there's something really special about that fertilized egg cell. And you may hear it said that, oh, it carries a blueprint for the organism. Well, I don't like that analogy because it's actually much more impressive, much more fascinating than that. There is no blueprint. It is a self-generative process, and each individual organism has to build its own body plan from a series of cellular and genetic interactions. And what is encoded in the genome is the capacity to set these processes in motion, but there is no set drawn out plan. And this drawing by Mark Escher, I think reminds me of this, because as the embryo is drawing its own body plan, it starts with a very rough outline, and then the detail gets filled in with time. So what's involved in controlling cell differentiation and spatial organization? Well, I don't want to get bogged down in molecules and details of molecular interactions, but just on a very, I suppose, concept level, just to introduce you to some of the main actors, some of the main role players in this. As cells become different to each other, they have, by and large, the same genome in their nucleus, so the same set of genes in their nuclei. But what's different is the genes that they switch on and off. So one of the main regulatory players in this are cell-specific transcription factors, so proteins that are able to bind to the regulatory elements of certain target genes and either switch them on or off. So those are really key role players in this. Other role players are involved in how cells communicate with each other and with their environment. So they encode the components of these communication pathways. So the signals and the receptors and the intracellular molecules that relay the message, and the ultimate response, which is usually the switching on or off of genes in the nucleus. So these are some of the key role players to keep in mind as we go forward. So what I'm going to do in the rest of this talk then is take you on a journey through some aspects of development, highlighted by some of the research that I've had the privilege to be part of. So I'm going to talk about how development is controlled, the genes that control development, the importance of space and time and how dynamic development is, how complexity is achieved in a complex vertebrate animal, and how we grapple to interpret that complexity, and why movement of the embryo is important. Okay, so first, a little bit of history, really, a history of how the genes that control development were first discovered, and the importance of the humble fruit fly, Drosophila, in that work. Well, the fly has a very obvious modular body plan, body structure, with different head segments with different shapes and structures on them, 3 thoracic segments, and each of the thoracic segments has a pair of legs, and the middle thoracic segment has a pair of wings, and then the abdominal segments that have a specific size and shape as well. And in a normal fly, structures like antennae and legs and wings form on particular segments. So any alteration in that pattern is very easy to spot. And not only in the adult, but also in the embryo, the fly embryo, the trained fly embryologist can see the telltale signs of these segments and see when that pattern is altered. So Drosophila mutant screens were designed to identify genes involved in forming this body plan. So by exposing the flies to mutagens and then looking for the very rare events of an alteration in the body plan, and then being able to work for that caused by mutations in a single gene, I should say, and then being able to work back from that to gain some understanding of how these genes work. So that big piece of work was undertaken in the fly. And one of the pioneers of this work was Ed Lewis, one of my personal heroes. And he spent many decades working on a particular type of fruit fly mutation called homeotic mutants. And here in this image, you see the head of a normal fly, and it has particular head segments. But notice these structures here, they're antennae, sensory organs on the head of the fly. And here's one of the mutants that Ed Lewis worked on. It's called the Antennapedia mutant. And in the place of these antennae, this fly has a pair of legs on its head. So a complete transformation of the structures on the head of the fly. And here's another example of these homeotic mutants. This is ultrabithorax. And in this fly, it has the normal pair of wings on the second thoracic segment, and it has an extra pair of wings produced on the third thoracic segment. So it's kind of a transformation of the identity of the third thoracic segment to be more similar to the second. So what these homeotic mutants show are at least a partial transformation of the identity of a segment toward another segment. And the finding of these single gene mutants really opened the concept that there are individual genes that can guide developmental events. And that these kind of genes are involved in directing the identity of body parts. So Ed Lewis, together with Christine Nusslein-Volhard and Eric Vischus, were given the Nobel Prize for Medicine or Physiology in 1995 for their accumulated body of work, which identified all of the genes needed to build the fly body plan. And it wasn't just because of the insight it gave us to how a Drosophila body plan is built, but as you're going to see over the next few slides, the significance of that to our understanding of the building of any animal body plan. So if those two homeotic mutations that you saw on the last slide, antennapedia and ultrabithorax, with the advent of molecular biology and researchers being able to study the genes molecularly and look at their DNA sequence, these two genes were of the most sought after genes to understand and to clone and sequence. And when they were cloned and sequenced, both genes were found to have a short DNA sequence within them that was almost identical across these two genes. And this was called the homeobox because it was found in homeotic genes. And this sequence showed that these gene products act as cell-specific transcription factors, that they can bind to DNA and regulate the switch on and switch off of target genes in particular cells. And if you've heard of Hox genes, these were the first Hox genes. But cloning and identifying these genes in a fly was the first step to understanding how all animals their body plans are organized, because it was quickly realized that if these two genes in one genome are so highly conserved evolutionary, then maybe there are similar genes in other organisms. So a search for similar genes in other organisms, in other animals, showed that these Hox genes are present in all animals with an organized body plan. And I don't want to give you the impression that Hox genes are the only genes that regulate the formation of the body plan. Those original mutant screens in Drosophila also identified other classes of genes. And these genes encoded components of cell signaling pathways. So the signals and the receptors and the relay mechanisms and transcription factors involved in cells communicating with each other and with their environment. And these are also highly conserved across evolution. So the genes that lay down the body plan of an animal are highly conserved over hundreds of millions of years of evolution. And all of this was revealed by that early work on the humble fruit fly. So how do these genes in vertebrates operate? And at the time, researchers were very eager to get their hands on these genes from other and study them in other organisms, and particularly the mouse, because at the time the mouse was one of the few vertebrate organisms where you could change a gene in a specific way and test its function. And among the people pioneering this work around the world was my very inspirational PhD advisor, Bob Hill, at the MRC Human Genetics Unit in Edinburgh. And Bob discovered a number of these Hox genes in the mouse. And my PhD studies were working with Bob, and in that we looked at two particular genes, and one of them we showed was switched on in a stripe of cells within the developing brain of the mouse. So this, just to orient what you're looking at here, this is you're looking at the dorsal surface of a mouse embryo, and you can see part of the brain developing here. And this is a section through that region, and the stain here, or the bright label here, is showing that those cells just in that region of the developing brain of the mouse have switched on this gene. And this gene is needed, or the expression of this gene in that part of the brain is needed to define that territory of the brain. Without its proper function, the neurons that are formed here do not make the correct projections and contribute to the system of building the brain correctly. So what we showed here was a correspondence between how this Hox gene in a mouse defines that territory of the developing brain of a mouse embryo, and how that corresponds to the roles of Hox genes in defining the body plan of the fly. Okay, so now I'm going to move to talking about how dynamic development is and how important it is to keep track of space and time when studying development in the embryo. So we work on limb development in the mouse embryo and limb development in the chick embryo. And so I chose out these images from Matt Kaufman's Atlas of Mouse Development to show you how dynamic and how quickly development of the limb happens in the mouse embryo. So the first image here is at 9.5 days post-fertilization. And at this stage, you see a little bump or two little bumps here on the surface of the embryo. And those are going to be the forelimb and the hind limb. And they're just mounds of proliferating cells at this stage. 2 days later, those limb buds, as they're called, have grown out and extended. And you now see a paddle on the end, and that paddle is going to be the digital plate where the digits form. And over the next couple of days, you see how the digits are starting to be visible. And actually the space between the digits is removed by those cells being directed to undergo programmed cell death. So in a way, the digits are carved out of that paddle by cells receiving instructions to be eliminated to separate the digits. By 14.5 days, so this is just 5 days later, there's a little limb, a little recognizable limb. Now, it's not functional. It wouldn't be able to bear the weight of even that early embryo, but it looks like a little limb. And these images that you've already seen generated in the lab are from this stage of development. So inside there's cartilage and bone and muscle and tendon, all differentiating, cells differentiating in the right spatial arrangement to make a mouse forelimb. So we're interested in how these shapes emerge and how tissues derive their particular identities, their distinct identities. So how cartilage is distinguished from bone, and the bone region is distinguished from the joint region, and muscle from tendon, and so on. And so what we know is that there are spatially and temporally regulated signals and cues that the cells respond to that influences their behavior and influences this spatial organization. And just as a bit of fun to show you as well, or to impress on you as well, how dynamic development is, this is a little movie that was made at the Edinburgh Mouse Atlas Project. And it's simply a series of sections through the part of the embryo brain where the eye will develop over 10 days of embryonic development. So I'm just going to play it for you first, and then I'll show it again and try to give you a little bit of guidance on what you're looking at. But I think it's nice to just look at it first. Et voila, in 10 days you have an eye. So just taking it back, and I'll show you some of the key features to look out for. So this is part of the forebrain of the mouse embryo, and these cells here will induce their neighboring cells on the surface of the embryo to thicken, to buckle inwards, and to form the future lens of the eye, while the cells derived from this part of the brain will form the retina. So watch out for that. So now, these cells are being induced to thicken, to buckle inwards, and this will form the lens of the eye, while some of the cells from the original forebrain extension will form the retina. So absolutely fantastic. And I hope you can see from that how we have to really keep track of 3D space and time when we're looking at developmental processes. And that's something we specialize in the lab, and we use a 3D imaging technique in order to be able to see the morphology of the emerging embryo, but also to see molecules located within the emerging embryo. So the products of gene expression. And so in these movies here, these are 2 mouse embryos, and they've been stained to show where different important developmental molecules are located in the embryo. And so we can look at molecular events within the morphology of the embryo, or we can look at emerging tissue morphology with cartilage in yellow here and blue for bone. And we've previously done a lot of work on the development of the lung buds. And to generate these kinds of images, we use a technique called optical projection tomography, which was invented by a very brilliant former colleague of mine, James Sharp, at the MRC Human Genetics Unit. And James developed OPT specifically designed to be able to capture vertebrate embryos. So the size of vertebrate embryos fits perfectly with this technique. And it also has another important feature that you can visualize molecular molecules within the embryo, as you can see here with the white staining in a mouse embryo or a chick embryo, or some work we've done also on frog embryos. And just to show you a little bit more how we capture this type of information, this is a photograph down the microscope of an 11.5-day mouse embryo, and the stained regions is using a technique to reveal where a particular gene is switched on, where that gene is active. The browny purpley stain. This is an OPT capture of that expression pattern in 3D. And now we can see the expressing cells in this white, gray tones. This is a true 3-dimensional computer representation of that embryo, so we can section it in any plane and look at the expression through the full depth of the embryo. And what you're seeing here is moving serial sections through the embryo, and we can see at any point through the depth of the embryo where the cells that are expressing that particular gene are. So this is the kind of data we can generate. And I'm going to move now from that technique into a piece of research that I've been involved with for many years, on and off. It's been an ongoing collaboration that stopped and started a bit, but and this is a collaboration with the Edinburgh Mouse Atlas Project. And I previously worked with the Edinburgh Mouse Atlas Project before returning to Ireland. And the Edinburgh Mouse Atlas Project is a true interdisciplinary project, and it's led at the front by a developmental biologist, Duncan Davidson, and a computational biologist, Richard Baldock. I started this project when I was there at the Mouse Atlas, focusing in on a cell-to-cell communication pathway, a particular pathway called the Wnt signaling pathway. And this Wnt signaling pathway is very complex. As I mentioned earlier, how a lot of gene discovery for developmental genes in vertebrates arose from the earlier work in the fly, and so too the Wnt pathway partially gets its name from a fly mutant called wingless. And you can guess what the phenotype of that mutation is. And also the Wnt signaling pathway has been shown to be altered and to be operating incorrectly in several different cancers. And there's massive complexity in this pathway. In the mouse and human, for example, there are 19 different genes encoding different Wnt signals. So the signal can be encoded by 19 different genes. The specific receptor that detects that signal is encoded by 10 different genes in human and mouse. And so with the groups of effectors and the transcription factors that communicate that signal through the cell. So there's multiple genes contributing to this type of cell-to-cell communication pathway. So what we set out to do is to look at the expression of all of the genes that are involved in Wnt signaling pathways, so more than 50 genes across multiple stages of development, capturing the gene expression patterns in the way I just showed you. So this is the when and the where of where the potential components of this communication pathway are switched on in the embryo. And because we can generate these 3D computer models, we had the ambition to be able to integrate all of these patterns into a single computer model of the embryo at any time. So making a detailed map of where all of these genes are switched on. So how to view the whole system of this cell communication across space and time, keeping track of the inputs, so the spatial and temporal positions and regions of where the inputs to this pathway are switched on, and also keeping control or monitoring the outputs. So the output of at least the major one of these pathways is the switch on of particular target genes. And so we have a transgenic reporter mouse that anywhere that a cell is actively responding to this signaling pathway, the cell will be fluorescent. So we can use that reporter mouse to capture where the pathway is actually firing, is active. So we're keeping track of the inputs and we're keeping track of the outputs. And this required a lot of data generation, and much of this data was the Trojan work of Kristen here. And so, more than 50 genes over multiple stages of development. And this just gives you a little flavor of the different gene expression patterns. You can see Wnt5a here is expressed in the limbs and the face. Wnt7b is expressed largely in the nervous system, as is Frizzle10. So very different patterns for each of these genes. Our next ambition then was, after collecting all of that data, to be able to integrate it and map it into an integrated representation, a digital representation, a three-dimensional map of where all these components are. And this turned out to be very challenging because no two mouse embryos are exactly the same. And you can see here that this embryo has its tail turned to the left, this one has its tail turned to the right. So this involved warping those data into a common space. And you had to be able to do this without distorting either the morphology or the data showing where the gene is switched on. And that turned out to be very challenging and took some time. But colleagues in Edinburgh Mouse Atlas were up to the challenge, and they developed this software called Wool's Warp that can achieve that. And we've spent a lot of time being sure that this process didn't distort the morphology or the data by checking everything back against the original data. So this just shows you what integrating those data look like. So here's the expression of one particular Wnt, Wnt1, Wnt5a, Wnt11, and here's those three patterns integrated in the mouse embryo. And this is the 19 different Wnt encoding genes integrated into the embryo on the extreme right. You can't see much detail there. So this movie shows the incremental adding of each pattern one at a time. So red is Wnt1, blue is Wnt2, green is Wnt2b, orangey is Wnt3, I think, I can't read that, Wnt3, Wnt4, Wnt5a in pinky and purple. So you can see each pattern being added one after the other. And what you can see is it's filling up the space of the embryo. So each territory is uniquely occupied by, not uniquely occupied, but there's a unique pattern of each gene that's filling out the embryo. And I like to call this the patchwork embryo. And it's going to keep going until all 19 are there. So we'll move on. And the other thing that the computer colleagues in Edinburgh did was they developed this web tool that allows you to interact with the data. So remember, these are true 3D models, so they can be sectioned and sliced in any orientation. And this allows you to move through any slice through the embryo, and you can switch on or visualize the expression of any of the component genes. So here we're just looking at the expression of Wnt1. Here we're looking at the expression of Wnt11 and Wnt2, and so on. So this gives a nice way to interact with the data and analyze it. But what has this brought us more than just looking at each expression pattern separately? Well, we can start to ask some questions we couldn't ask in any other way. This is showing where each of the Wnt genes is expressed, but this here is a union of all of those domains. So it's saying where any Wnt signal is being switched on in the embryo, what territory that is. And here is where 0 Wnt are detected. So we couldn't ask that without having the complete set of patterns. Here we can ask where multiple Wnt genes are expressed together in the same territory of the embryo. So revealing kind of hotspots of where multiple Wnt's are expressed. And you see these hotspots in this particular embryo here. And some of them are areas that we already knew were really important in development, like this part of the forebrain, for example, part of the neural tube. But some of the areas are areas we had no idea were important for signaling before. This one here, we like to refer to as the armpit, because it's located right where the limb meets the trunk of the embryo. And you can see massive activity of Wnt signaling genes there that we weren't aware of before. And this is we're now seeing these hotspots in the whole embryo in a different way. And here on the right in green, you're seeing that activity of the pathway, the output of the pathway that I explained. We use this transgenic mouse where the cells become fluorescent if the pathway has been activated. And there's quite good correspondence between the two, but not perfect. So here in this part of the brain, we can see there's a lot of Wnt signal activity, gene activity, but there's no pathway activity. And on the other hand, this part of the forebrain here that we've looked at quite closely has a lot of pathway activity, a lot of firing of cells responding to Wnt signaling, but has very limited expression of Wnt pathway genes. And this is really just making the same point. This is looking at the limb bud, and we can name these hotspots, give them names of according to their position, and we can superimpose on that then where the cells are responding to a Wnt signal. And again, we see the same thing, that some regions have switched on a lot of genes, but they aren't actively firing at that time. And here there's an overlap, but it's not a complete coincidence of the two sets of cells. Okay, so now I'm going to move to a new concept and talk about why movement of the embryo is important. So vertebrate embryos move a lot from early in development. And why is that? So here you see some fish embryos, and this video has been speeded up, so they don't move quite this fast, but they do move a lot. The second video you're going to see here in the middle was made by an undergraduate student, and he was examining or comparing the rates of movement and how chick embryos move inside the egg. So you can see the limbs moving, the trunk moving. So this is videoed inside a developing chick egg with a window cut in the shell of the egg. And on the right is a 4D human fetal scan with the well-known movement of the fetus in the uterus of the mother. So all vertebrate embryos move from early in development. And why do they? Why do they move so much? Well, we know that mechanical stimulation is very important for maintenance of the skeleton. So, for example, when astronauts go into zero gravity, they lose bone mass. Tennis players in their dominant arm will have increased bone mass there. So maintenance of bone responds to mechanical stimulation, but very little was known about how formation of bone and skeletal structures in the embryo respond to mechanics. But there were indications that it was important. And this study in the 1980s looked at a lot of clinical cases where the thing in common between all of these newborn infants is that they had reduced fetal movement for different reasons, some of them because maybe reduced amniotic fluid or because of neuromuscular disorders. So various reasons why they moved less, but the thing they had in common was that they moved less. And they led to a spectrum of skeletal defects, including brittle bone disease, even leading to fractures under normal birth. And joint deformations, changes in joint shape, and the most well-known of those, hip dysplasia. So this was an indication that these movements are very important for the formation of the skeleton. So we set out on a journey to look at that more systematically, and in a collaboration with a bioengineer, Patrick Prendergast, we took a two-pronged approach. So we immobilized mouse and chick embryos, or we examined the effect of immobilization in mouse and chick embryos. In the mouse, we could use these mouse mutants that develop without muscle or with immobile muscle. So we could look at the formation of the skeleton in the mouse embryo in the absence of muscle movement. In the chick, we could do something completely different. We could administer a drug that prevents movement in the egg and look at the effect on immobilization. So very different types of immobilization in two different species, and then examine the effects on bone formation and joint morphogenesis. And then in parallel with that, under the direction of Patrick Prendergast, we could capture the realistic morphologies of the emerging skeleton using our OPT imaging, and then use that to inform simulations of movement and prediction of the types of biophysical stimuli that are being generated and where, and the patterns of these stimuli. And comparing these two sides of the study, we saw a very interesting correspondence between particular types of biophysical stimuli and the patterns of stimuli, and where we could see the changes in the development of the skeleton. So this is summarizing about 10 years of work in this one slide, or these two slides. So to summarize, what we found was that in both the mouse and the chick with different types of immobilization, that bone formation was delayed and abnormal, which reflects the brittle bone phenotype seen in these newborn infants, and joints were abnormal. So here in a control chick embryo, you see the dark staining is where the bone will form, and in between where the joint tissues will form. And in this immobilized embryo, in extreme cases, you get a complete fusion across the joint line so that the skeletal elements become fused across the joint. We also showed changes in the shape of the skeletal rudiments as they're forming. And so that led us to propose that these biophysical cues act as a kind of positional information that guides the formation of tissues in the right place. But we didn't know what the underlying molecular mechanisms were, and we've spent a long time now trying to figure that out. So what kind of molecular cues could be generated by these biophysical stimuli that the cells are responding to? And we haven't figured it out yet, but we're getting somewhere with it. So we first took an open-ended search of what kind of communication systems are altered in immobilized embryos, and we found our old friend Wnt is disturbed and another pathway called BMP. We looked at a particular type of signaling that's known to be important in how a cell responds to its environment called hipposignaling, and we showed that it's active in the regions where the bone shape will change that's altered under immobilization. We looked at a little structure that sticks out of the surface of many cells called a primary cilium that can act as a sensor of the biophysical environment. And we showed that these, and this is Claire's lovely imaging of one of these cilia, these finger projections on the surface of a cartilage cell within the developing limb. So we've shown that these primary cilia are in place, ready to sense the mechanical environment. And probably our most revealing finding was in this paper we published in 2018, where we showed how the signaling at the joint changes under immobilization. So in the normal embryo, Wnt signaling is, so these cells here are at the future joint that will form the articular cartilage at the joint. They're receiving a Wnt signal and they're responding to that Wnt signal. That's shown by these red dots. At a distance, the yellow dots are showing where the cells are receiving and responding to a BMP signal, where they will form bone. So these cells will form bone and these cells will form joint cartilage. In the immobilized embryo, the Wnt signaling at the joint disappears and the BMP signaling is continuous across the joint. So the territories that will form a joint and will form the bone are being misguided because they don't have the biophysical stimuli to form the correct tissues at the joint. And a last little study in this bit that I want to tell you about, where we looked at if joints can recover if movement is resumed. So we did a study where we immobilized the embryos over a period of time, and we compared that to immobilizing for a shorter period of time and then letting the embryos recover and start moving again, and compared that again to the shorter immobilization, and then followed by hyperactivity. We could stimulate the embryos to move faster. And then we looked at the effects on the joints. And to cut a long story short, there's partial recovery and better recovery at some joints than others, but the hip joint particularly recovered very well. So this is our control situation where in 100% of the embryos, we have these nice cavities forming at the hip joint. In sustained immobilization, 100% fusion at the hip joint. And particularly where there's immobilization followed by hyperactivity, we get only 1 out of 8 cases with a fusion. So there is recovery in the formation of the joint, even if there's immobilization, but it's followed by hyperactivity. And so this has implications for the possibility of appropriate physical therapy for infants or for fetuses that have reduced movement. Okay, so the last thing I want to tell you about tonight is some of our current work. Again, looking at the importance of movement, but shifting our focus now to the tendons. This work is in collaboration with Spencer Chesney at Penn State University, who is a biomechanics or bioengineering expert there. And to give you a little bit of background of why we've decided to focus on the tendons, well, they have a very interesting and very complex structure that's very important to their function. The cells of the tendon will form extracellular proteins and complexes that give the tendon its important, strong, fibrous connective tissue that allows it to transmit forces from the muscle to the bone. They're very prone to injury, which you're probably aware of, especially in athletes. And repair of tendons is very limited. They can be repaired by surgery, but it always leads to a tissue with reduced strength and stability and prone to re-injury. And there's been no successful attempts to date to regenerate tendons in a dish. So some of the earlier work in the lab acts as a grounding to this. So we're looking at the appearance of different tissues in the chick limb. And you can see that the green label here is where the tendon cells are differentiating as early as embryonic day 8. So the tendon cells are differentiating in the right position in the limb very early on, but they don't have the functional characteristics of a tendon. They're much softer than a tendon should be. And we know from other data that, from other work, that there's a rapid transition in the structural and biomechanical properties of the tendon that happens between embryonic day 16 and 17. And shown here in earlier work by Burke et al., in the length of collagen fibrils extends enormously at embryonic day 17. So we wanted to zone in on this maturation and learn what's involved in directing this maturation in the embryo. And we wanted to examine it mechanically and structurally. So in Spencer's lab, this is the work of Ben Peterson there, they've devised a way of being able to test the mechanical properties of these tiny little embryonic tendons. And this shows that, so the green is embryonic day 16, 18 in orange, and 20 in blue. And the take-home message here, and this is looking at the stress experienced in the tendon as a function of the strain applied. And there's an enormous increase in the stress-strain relationship between E16, embryonic day 16 and embryonic day 18. And that's again reflected here with a parameter called modulus, which is about the stress-strain, or which captures the stress-strain relationship. So you see this big increase at that stage. And then in Dublin, we've been looking at the structure of the embryos and showing that there's a big step in the size and the spacing of the tendons and the alignment of the fibrils, the collagen fibrils over this time as well. Then what happens in immobilized embryos? Well, in immobilized embryos, and you see blue is control here, and orange and purple are two different types of immobilization. This increase in the mechanical properties that we see in normal development is gone, is lost in immobilization. And also structurally, immobilized tendons resemble earlier tendons. So that transition over development is gone. So the step change that we see over late development is lost. So we're now examining the molecular mechanisms involved, what kind of pathways could be interpreting these mechanical signals, and important for the maturation, the normal maturation of tendons. And we're growing tendons in a dish. So this is the work of PhD student Grace Morris, and Grace she puts these pins in a small culture dish, and then she makes cells from the early chick embryo tendon and isolates them and then plates them in a fibrin gel. And the fibrin gel creates a kind of natural a more natural three-dimensional environment for the cells. And in those conditions, over time in culture, these cells contract and self-assemble, and they pull this whole 3D culture between these stress points that are created by the pins into a rolled up structure like a tendon over, this is 11 days in culture. But how similar are they to tendons? This is a section through an embryonic tendon and a section through one of Grace's tendons in a dish. And we can see that the cells have aligned nicely and that they're making this extracellular matrix of collagen as a tendon should. But we don't expect them to go through that maturation process in the dish. But what we have here is an experimental system that we can then test out our ideas of what we're learning in the embryo to see if we can stimulate them to undergo maturation. And this work has now been expanded with a wider collaboration with a view to translational work. So with a group that works on gene delivery using nanoparticles and biomaterials. So really with a view in the future to being able to construct tendons that could be used in preclinical trials. And this partnership is funded in Ireland by Science Foundation Ireland and in the US by the National Science Foundation. So my last slide is the set of images I put together to persuade the funders and the reviewers of what we were trying to do here, and it just sort of encapsulates the idea of this research. So in the embryo, we want to discover the mechanisms that build a functional tendon. In these tissue engineered constructs, we have a system where we can test out those ideas and hopefully build a more structurally robust tendon in a dish. And then ultimately, before the end of the project, to test out some of those tendons in small animal models. Now, this part of the work is our collaborators' speciality. We won't be involved in that. But what we're doing is discovering from the embryo and trying to apply it to making tendons in a dish. So I hope you can see that's where the title of today's talk came from, learning from the embryo how to, in this case, build tendon. And I leave you with another quote, this time from Scott Gilbert, a developmental biologist and prolific communicator, who wrote in 2010 that understanding development promises to expand medical capacities in the present century as much as microbiology in the last. And he's inspired to say that from how developmental biology is increasing our fundamental understanding of cell control and the applications to, for example, tissue engineering and stem cell therapies or cancer therapies on the one hand, and increasing our understanding of birth abnormalities and their management, especially in the more challenging environments of today's world. And I'll leave it there and answer any questions you have. Just with thanks to very many people. I've mentioned some as I went along, but funders and past team members. Rebecca, who did a lot of this work, now has her own faculty position in Trinity, and we still collaborate. Past collaborators as well, and our present collaborators, the Edinburgh Mouse Atlas Project, and for the Tendon Project I told you about just at the end. So thanks very much for your attention.